Hello everyone, my name is Alejandra Garcia and I am currently doing my PhD at the University of Vigo. Today I'm going to share with you part of my research focusing on how we study the physiological response of an aquatic invasive snail using proteomic tools. Nowadays, invasive species are one of the biggest threats to global biodiversity. Freshwater habitats are especially vulnerable to biological invasions due to their close connection with human activity and for having fewer natural barriers. As a result, freshwater biodiversity is declining much faster than in terrestrial ecosystems. The New Zealand mud snail, Potamopargus antipodarum, is one of the most widespread aquatic invasive species worldwide. It's very small, just 4 to 6 millimeters, but highly adaptable. Although it's tiny, this snail can have a big ecological impact. It disrupts food webs and outcompetes native species, sometimes even replacing them. It's also highly resistant to predation, even surviving digestion by fish, which can alter the diet and nutrition of predators. So why is it so successful? Its ability to reproduce quickly and asexually combined with the absence of natural predators and its tolerance to harsh conditions like extreme temperature, salinity, and even periods of dryness gives P. antipodarum a clear advantage as an invader. But among these traits, we are especially interested in one, its tolerance to salinity. Why study salinity? Its adaptability to different salinity levels likely plays a key role on its success. It helps the snail survive long-distance transport in ballast water and establish population across a wide variety of habitats, from freshwater to brackish environments and even coastal areas near the sea. For this reason, our main aim was to understand how P. antipodarum adapts to different salinity conditions. To do this, we carry out salinity tolerance experiments combined with a proteomic analysis to uncover the molecular mechanisms behind this adaptation. We first collected around 300 snails from a river in northwestern Spain and established cultures under controlled conditions. Then, after an acclimatization period, snails were exposed to 5 salinity levels, from freshwater as the control to full seawater over 28 days. We measured the effects of increasing salinity on survival, activity, and reproduction, including embryo production, newborn viability, and size at birth. So what we found? We observed higher survival rates than in previous studies. Survival remained at 100% up to salinity of 20, it dropped to 80% at salinity of 30, while in seawater all snails died by day 5. In terms of activity, snails didn't move in seawater. However, at salinity of 30, activity was initially low but gradually returned to normal after about 10 days. High salinity also impacted newborn survival and development. Mortality increased in seawater, but surviving juveniles had smaller cells, suggesting that salinity stress could induce premature birth. Interestingly, embryo production remained consistent across treatments, even at high salinity levels. This is a novel result, as previous studies reported a sharp reproductive decline beyond 15 PSU. Looking at these results, we wondered about the molecular mechanisms that enable this snail to survive and reproduce in both freshwater and salt environments. To explore this, we conducted high-throughput proteomic analysis. In this case, we only used 2 salinity treatments, 10 and 20 PSU. We carry out 2 exposure durations at 24-hour exposure and a 7-day exposure. We extracted proteins from snail samples, then we made the digestion into peptides using trypsin, and using TMT reactives, we labeled each sample with an unique chemical tag. Then the samples were analyzed using liquid chromatography and mass spectrometry. And to identify proteins, we create a database by combining published transcriptomes of this species. Finally, we analyzed the proteins that were differentially expressed across salinity treatments with bioinformatics tools. We found that this snail modulates its protein expression in response to salinity. Here, in this volcano plot, we can observe that the most changes occurred under high salinity and longer exposure, while low salinity had no significant effect in protein expression with short-term exposure. When we looked at the potential functions of these proteins, we saw proteins related to amino acid metabolisms, suggesting the activation of free amino acid synthesis to maintain osmotic pressure, also shifts in metabolism, oxidative stress responses, immune and defense mechanisms, and cytoskeleton regulation. These findings suggest that this species possesses osmoregulatory mechanisms that allow it to acclimate to changes in water salinity. However, more detailed analysis is needed to fully understand these responses. Salinity tolerance is a common trait among aquatic invaders. For example, the blue crab tolerates salinity from 0 to 65 pSu, and the red swamp crayfish expresses ion transport genes under salinity stress. This kind of physiological flexibility gives invasive species a competitive advantage over native species and contributes to invasion success. As human activity and climate change continue to alter aquatic environments, understanding how invasive species respond to environmental changes is key to predicting vulnerable areas and to develop prevention and control strategies. Thank you very much for your attention. I'll be happy to answer any question or comments you may have.